T-Past. So welcome, Hans Blom. Hans Blom did this postdoc and quite a lot of research with Stefan Hell, who is one of the Nobel Prize winner for super resolution microscopy a few years ago. And he also has developed a lot of STED microscopes and set up super resolution at KTH here at the Royal Technical Institute or something like this in Sweden. and he now manages the advanced light microscopy facility at SciLife Lab, including but not only super resolution microscopy. Please, Hans.
Okay, yeah, thank you, Sylvie. So when you say not so long ago, it's actually over 10 years ago since the Nobel Prize, and it's
right.
over 20 years since I was a postdoc in Stefan's group. So yes, it's okay. So I will give you a small introduction here to super resolution fluorescence microscopy. And as Sylvie said, I'm at KTH, the Royal Institute of Technology, but I'm managing the advanced light microscopy facility at SciLife Lab. So all the techniques I present, you can get support with in our national facilities. So just contact us. So let's then start with, Sylvie said here, there was a Nobel Prize a few years ago regarding better resolution for fluorescence microscopy. And the prize was actually awarded in chemistry. One would maybe think that microscopy is a tool more close to physics. But actually, as you know, in fluorescent microscopy, you need to label your sample, you use fluorescent molecules, and there were a couple of tricks with these techniques that was awarded the Nobel Prize that actually manipulated the fluorescence state of molecules. So that's why they gave it to chemistry. So yeah, I will go through both of these techniques that was awarded the Nobel Prize. So here are actually the techniques I will cover in this short introduction lectures. So to the left, you see what is called single molecule localization microscopy. That was a technique that was awarded the Nobel Prize. And there you detect individual single molecules and you pinpoint their coordinate. And you can get quite high resolution. That's a wide field method or ILO method, turf method, kind of illuminated close to the surface. And you use a sensitive camera to detect fluorescence. Then, as Sylvie said, Stefan's technique was dubbed STED. It stands for stimulated emission depletion. I will go through that process too. But basically, it's a confocal-based microscope where you use an extra laser to turn off fluorescence in parts of the focus. And then you can, so to speak, scan up an image there. And those two techniques gives you tens of nanometer resolution. I have ballparked some of the numbers there that you see, maybe down to 25 or down to 50 nanometer with these techniques. Then there are several other super resolution microscopy techniques, or maybe one could dub them soft super resolution or whatever you want to call them. And one is to illuminate the sample with stripes. So this is the SIMAC, structure illumination microscopy. And by illuminating the sample with a stripe pattern, there is a, so to speak, an interference with the spatial pattern of the illumination and your structure in the sample. And there you can actually down convert higher frequencies that the microscope normally doesn't see with the continuous or homogeneous illumination. And that's also a camera-based wide field method. And then Sylvie wanted me to mention shortly about one of the other soft super resolution microscopy techniques where you actually use a kind of a ray detector that use the size of the pixels there as small pinholes. And there you can increase the resolution a bit from the wide field microscopy too. So let's then start with just saying that this is the only parameter we are going to talk about in this lecture, that is to improve resolution. And then, of course, we need to kind of have a measure for what is resolution. And usually in this microscopy course, you probably talked about like point spread function, or how big is the image from a single molecule, and you have these area rings, or the separation of these peaks. And if the object then comes closer and closer, these peaks, they kind of start merging, and then you cannot separate things if you're trying to separate two of the same. Of course, if you would have different spectra here, one green and one red, then of course you could probably still see them in individual channels and things like this. So when talking about resolution, you usually talk about this possibility to separate the same object. And then you see here like the width of this point spread function. That's kind of also a measure of how sharp is your pencil painting this image, you can say. So we are only going to talk about resolution as parameter. And you know there are several other parameters in microscopy that are of importance. So, but let's then jump on Stefan's method instead. And he was actually a postdoc 30 years ago in Finland. And then he got this bright idea that, hey, in a fluorescence microscope, we can usually not separate the molecules in the focus, because the focus is kind of large compared to the molecules, usually a few hundred nanometers wide, and 3 to 4 times larger along the optical axis. So those 5 molecules in the focus there, we cannot really separate. And then Stefan had the idea, writing this theoretical paper, let's build this microscope. Let's build a microscope that turns off the fluorescence of molecules everywhere except in the voxel where we, so to speak, want to measure. And then we can scan this around and build up an image. So how is this now possible to be able to turn off fluorescence like this? Well, the book he was actually reading as a postdoc when he got this idea was about stimulated emission. So there are a few processes when you excite fluorescent molecules that goes about. You shine in some green light to a molecule, the molecule absorbs this light, and it gets excited, and then after a while it relaxes. And after a while there's usually nanoseconds, and out comes an orange photon. And that's the process that you're using in fluorescence microscopy. You use stimulated absorption to excite the molecule, and after a while it spontaneously relaxes and sends out the fluorescence signal. But then already 100 years ago, Albert Einstein told that if you can excite a molecule, that is called stimulated absorption, you can equally well de-excite an atom, and that is called the stimulated emission process. So it's basically the stimulated absorption is somebody kicks you and you get excited up on the table, and if the de-excitation part is like a hammer hitting you on the head, and you fall down on the floor again. So in the lower graph there, after you have excited the molecule, you can de-excite it by sending in a little bit of redshifted light, and out comes then a copy of this energy that is carried away. And we don't detect that red photon. We only wait for the fluorescence, the orange photon in this case. So then here is the principle of a STED microscope. In a normal microscope, you have an excitation profile, and you see all those stars up to the left. But if you now add the red profile and you create it with a, so to speak, zero intensity in the middle, and the higher intensities are now going to have the stimulated process de-exciting those molecules. And then we sit and wait for the fluorescence. And then we increase the steady intensity quite a lot, because we only want to have fluorescence in the very, very center of this profile. And then we scan this co-aligned excitation, the excitation focus across the sample. So it's a confocal microscope with an extra laser. So you see a typical absorption spectra there where you excite with the laser, and then you select the depletion laser far out to the right, so you can still detect a lot of fluorescence across your bandpass filter there. And if you then schematically just look at the buildup of these microscopes, you have a normal confocal microscope, you have a laser excitation, and then you have a pinhole on a sensitive detector. And then you add that extra beam path for the sted, and you shape it in this case as what is often called a donut. And then you crank up the power of that one, and now you can separate all those molecules that you previously could not separate. And then you scan that image. Does it work? Yeah, well, if it didn't, we had to tell Stefan to return his Nobel Prize, but it actually worked quite well. So here you look at mitochondria and cytoskeleton proteins. You have the confocal reference above. That's just when the sted donut is turned off. And then when you turn it on, you get sharper images, and you can then localize the proteins sitting in the mitochondria with about 50 nanometer precision resolution. It's also possible to do this along the optical axis. You create then a donut that depletes above and below the normal focus. So then you shrink the height. And if you then combine shrinking both on the height and also the width of the focus, then you can get a very nice 3D resolution. And again, you see mitochondria there. And you don't really resolve the upper and the lower membrane in a confocal microscope. And the labeling here is just immunolabeling, and then you have selected a dye that is suitable for the wavelength to excite and deplete. So you can use like normal dyes like Alexa 488 or Alexa 647, but there are also newer dyes that have been synthesized to be more focus stable and work perfectly for sted microscopes. So here is a short summary then of what you get with sted. You get 4 to 5 times better resolution. You can also do this in 3D. Multicolor is possible. I wrote here 3 to 4 colors easy. Well, the easy part actually is complex because sample preparation with multicolor is not too easy to start with. But we have several excitation wavelengths and several depletion wavelengths of the systems. So 3 color is easy. 4 is probably also easy if you can make the samples. But usually we start with 1 and 2. So you can use your old cell labeling protocol, and then we probably guide you to select a sted dye that is Formula 1 dye that can give you even better confocal images to start with. Regarding speed, that depends of course on the field of view, but usually you zoom in on part of the cells to take your image. So then it goes as fast as a bigger image with the confocal. Because of course now you need to step with the pixel size of about 20 nanometer instead of like 60, 70, 80 nanometer. And of course, when you then shrink this focus, in a large focus, you maybe have 20 dye molecules giving you the signal. Now you make that 5 times smaller in each direction, so you're only having a few, maybe even a single dye molecule in the focus. So you're going to be a little bit contrast limited. Live cell is possible. It's of course challenging just by labeling live cells. Fluorescent proteins are usually hard to sted because they're not that photostable, but you can then use, of course, nanobodies against fluorescent proteins and you have a sted dye on top of it. So let's see. Yes, let's then switch gear to the other part of the Nobel Prize, which was awarded to single molecule localization microscopy, or 20 of the other acronyms that are out there in this field. And here you see a schematic overview of how the method works. So if you now could have just a single molecule sitting in your focus, the emission of that one would of course be your point-spread function. And then if you detect that on the camera, you kind of see a blob on the camera and it has an intensity profile. And if you now do the mathematical assumption that, okay, if we can pinpoint the center of gravity of this detection spot and assume that that's where the molecule is sitting, we can, of course, increase that precision. And the better signal we have, the smoother signal we detect, the more photons we're detecting from the molecule, the better we can localize this. So here you can then go from a width of about 240, 50 nanometer down to maybe a precision around, below 20, or depending on what method you're using. So that was the method that Eric Betzig and co-worker used in 2006. They made this experiment. So what they actually did here, they took activated fluorescent protein. So they used a photoactivable fluorescent protein. And then they used a pulse of UV to just stochastically activate a few molecules. So first you couldn't see the sample, you then activated the sample, and after that you excited it with the laser to measure the green fluorescence. And on the camera you saw a few molecules being on, so to speak, blinking. And after a while those were bleached, and then you repeated this 300,000 times or something. So the first time they made this measurement, it took like 9 hours. But today it takes tens of minutes. So here you then see a film from the camera that we did many years ago. So we used the same photoactivable fluorescent protein, shined in a little bit UV, and then the molecules that got turned on were excited and emitted the fluorescence. And here you see the molecules being on a few camera frame there, and then they disappear, and then new ones reappear. And we're actually detecting inside neurons here, or on the plasma membrane of the neuron, there's sitting a protein called sodium potassium ATPase pump. And that's the topology that they are pinpointing. And in this case, the microscope was in the turf mode, so the excitation was only at the very surface, just a few hundred nanometer into the sample, and it was detected on a sensitive CCD camera. So after you now film this for 20 minutes, recorded several hundred thousands of events, you then do the mathematical processing of finding the center of gravity of these blinks. And then you can plot them as a synthetics map up to the right there along the dendrite, and you see these spine structures sticking out. And as a counter staining, we have the postsynaptic density label there in wide field. It was basically to find the sample, because the photoactivable GFP is not present before being activated. And now with single molecule localization microscopy, you get kind of a map where are your events detected, and you can maybe start counting and quantifying these with different measures. How many molecules are there? Do they form like clusters or things like that? And actually there are also possible to get dye molecules to blink. So you don't have to use fluorescent proteins, which are, you can of course clone into your cell. So you can actually get dye molecules to blink. And the trick here is actually to shine a lot of laser light to the dye molecule. And you will then turn them into what is called a dark state. It's kind of a radical states. And then you have buffers in the solution that, so to speak, can dequench these out of the dark state. So then you can start blink. And you see some blinking molecules there where we have labeled tubulin with an antibody, and on top there the dye is Alexa 647, and then there is a buffer solution in there that you get the blinking to work very well. And this technique was invented at Harvard by Xiaowei Xuan, and in Germany around Marcus Sauer and Mike Hyland and several other scientists, just flooding the sample with a lot of light and then getting it to blink. So schematically you see down there how one then per frame localize individual molecules, and then you can build up a synthetic super resolution microscopy image. And a third method that is quite popular in these days regarding single molecule localization microscopy is to use what is called DNA paint. So to the left there at the top, you see microtubule labeled with an antibody. And on the antibody, there is a docking strength, a DNA docking strength. And in the solution, there is an immature strength that is then complementary DNA. And then through binding, you can generate on-off blinking. So when the immature strength is just swimming around in the solution, it moves quite fast. So you don't get much photon per pixel. But then you have a binding event, and the presence then sits there for a while, a few tens of milliseconds or even up to 100 milliseconds, and then you collect the photons and can localize the structure. And then, of course, you need to repeat this 10,000 times or maybe even millions of times. So you can take a little bit longer time here now because the binding is a little bit slow. And then, to the right, you just see an example where a French group used this, what is called DNA paint. You use DNA probe to paint the nanoscales, so you can then get a hint of how many synaptic proteins do you have in the spine structures. And I haven't told you actually what you should use this method for. Well, you should think about something small. And that's, of course, easy because everything kind of in the cell is small. So that's the nanoscale. So if you want to have a better image of your favorite organelle or whatever structure you're after, you can apply this method, single molecule localization microscopy method, or the STED method, and crisp up your image hopefully a bit. And then here is just a summary of them single molecule localization microscopy. You get to 7 to 8 times better resolution. You can also do this in 3D. Again, the field of view is a little bit small compared to a confocal large overview, but that's because it takes a while to blink all these molecules inside this field of view. And you also want to have a good homogeneous illumination so everything blinks the same. So the speed will be quite slow. It takes a while to blink this image. You need to have, of course, a little bit of a special labeling where the molecules needs to be switched. So there are several good molecules that can do this. And of course, you need good contrast. You cannot sit on any background. The easiest way to understand that is just to look outside and count how many stars do you see in the day. You can get the low background, you're going to see your single molecules blinking very well. And then, of course, pinpointing that center of gravity is a little bit model dependent. But today there are several very, very good algorithms doing this very fast, also open source. So then I will switch to these more soft super resolution microscopy or whatever to call them. So this is the first one, SIM. So if you have a sample, and imagine now you cannot see the structure in this sample, but for obvious reason I have drawn it so you can see it. If you now put the illumination in the form of stripes on top of this, you're going to generate what is called Maré pattern, an interference pattern, telling you about the structures with this striped illumination. And now you see that you have very broad, coarse lines. So they are easy to separate by the microscope. In the sample itself, it was very, very hard to see the very fine structures. But now we have, so to speak, down-converted higher spatial frequencies that can easily be detected by the microscope. But of course, the images is distorted or overlaid with illumination. But if we now know how we illuminate it, what spatial frequency we have in the illumination, we can, so to speak, process that part out of the image and then get back to a higher resolution in the sample. So this is basically how SIM works. You're illuminating your sample with these stripes, and you have the pattern rotating in the sample, so you get good resolutions in X, Y, and Z. And then you use this processing method to extract the information of the sample. And then you can crisp up your image with about a factor of 2 compared to wide field confocal. And it has also good 3D optical sectioning. And if you think about how this SIM process actually works, a microscope detects parts of what is the spatial frequency of the sample, but it can only see up to a certain threshold. And I think Sylvie will actually after lunch talk a little bit more about Fourier transform and Fourier spectrum and things like that. So if you now add this striped illumination, then you have actually a new illumination frequency. that, so to speak, carries information from the higher frequency that was previously not detected in the microscope into the microscope. And then the last step, you then have to put that back where it actually came from. So that's the mathematical process. So one of the first inventor of this technique was Mats Gustafsson, who was actually also working at KTH, but then he moved to the United States and worked there for several years. So if you then create a microscope where you have periodic excitation pattern, that is the stripe pattern, you have them in all 3 dimensions, then you can get this SIM method to work. And you can also have other periodic structures, lines and dots and things like that. And here is just some results that I think Mats took many years ago. So the wide field blur out of focus is then crisped up optically here with the SIM method. And then, as you already discussed, image processing and denoising and things can, of course, also in the end then help this image to become more beautiful. And I think after lunch, Eric will talk about deconvolution processes, so you learn more about the imaging analysis part. And regarding imaging analysis, there are actually processing algorithms emerging now in the last few years where they process SIM information down to 60 nanometer precision resolution. So it is possible actually, if you have a very good image and a good algorithm to process these soft super resolution images down to steady resolution. So here is then a small summary of SIM. The classical version gives you 2 times better resolution. You get optical section in 3D. Multicolor is pretty easy. It's camera based with filters. You have a slightly larger field of view compared to the hard super resolution methods. You can use your protocol that you're used to and the dice, but of course, learning about better dice, you should also always apply them. We're learning about better fluorescent proteins. You should switch to the most modern one, of course, because then you also get a better wide field image or focal image. SIM is slightly, of course, slower than the wide field method because you need to have those striped moved over the sample in different directions. So you need to generate like 10, 15, 30 images. That, of course, slows it down. But this pattern can be moved very fast. And again, of course, you need good contrast, especially as there will be processing step in SIM, so you don't just calculate out noise. And live cell is, of course, possible as it is a wide field method. But then, of course, the need of several images will, of course, slow it down a bit. And then I was asked to cover the last soft super resolution method that has a lot of different names. And that is, in principle, in a confocal microscope, you can actually get much higher resolution than you're probably used to by just closing down the pinhole. So if you have a fully open pinhole or a larger pinhole than what you're kind of used to set it one area, but of course you're going to see a lot of out of blur emission. So then you close down the pinhole, the optical section is improved, the resolution is also slightly improved. But then you close down the pinhole a lot. Maybe you close it down to 0.3 area unit or so. And then you can actually increase the resolution a bit. But of course, the contrast is going to die. You don't get any light through, so you're going to get a very, very, very noisy image. And many years ago, Colin Shepard, he actually was kind of one of the modern fathers of confocal imaging. He wrote an article, if you now move the pinhole slightly offset, that means that only part of the pinhole is, so to speak, overlapping with the excitation, then you get a smaller pinhole, and you will then increase the resolution in the microscope. But again, you would of course sit on this problem of that the contrast would become lower. And I don't know if Colin ever wrote a patent about this, but of course it was so long ago that 20 years later that patent would have run out. So then in the last 15 years, there's been a lot of development on having small pinholes on arrays and selling microscopes. So you see a lot of acronyms out there: AirScan, imaging, scanning microscopy, pixel reassignment. And I think Sylvie will have some questions for us to discuss later too. And then, of course, the other problem, 40 years ago, your computer could only save 0.5 megabytes. So if you now dreamed up this experiment, you take small detectors acting as small pinholes. They will improve your resolution. But each of these pinholes, as they are very small, these camera pixels are very small. Individually, they will detect very little light. But if you now have an array of these, then you're going to detect all of the points per function. But now this is from the size area detector. You have 32 of these. So now you're going to have 32 more information than before. And this could not be realized 30 or 40 years ago because there was no computer power. But today this is simple. To have a small array of detectors, Nikon call it Spark. You just saw a nice image in neurons from the AI talk. And what you actually then need to do is, of course, the shifted pinhole will not, so to speak, have the information of where you're exciting. So it will be slightly shifted, the image. And then you need to reassign that to its right position. So that's why it's also dubbed pixel reassignment microscopy and things like that. But the image will have a higher resolution. And now, of course, detecting on 32 places, in this case for the size detector or whatever number there is on other systems, you of course collect a lot of light. So then you can have this kind of system. If you normally have a confocal with just one pinhole, you close the pinhole down. Actually, if you have a very good sample, you can increase the resolution a bit. The contrast will become lower, but maybe then you can use this AI denoising method and get it back up to signal again. Or you use these small array detectors on the NSPAR or the area detector, and then you get soft super resolution microscopy. And if you need help, as I said, with some of these STEM technique or SIM techniques or single molecule localization microscopy technique or area, or you can ask Sylvie for the NSPARC technique, you just contact me and Anna, who is doing all this imaging. And we are at the SciLife facility. And we are a national facility, so anybody is welcome to come and get this support. And then there are two methods that I actually haven't covered in these lectures that you probably heard about. So expansion microscopy is that you kind of stretch your sample and thereby increase the resolution. And this is now a 10-year-old technique. And we have expertise on that part too. So you can learn protocols from Anna and you can stretch your cells. The downside of this one is, of course, if your cell now becomes several 10 times bigger than before, then of course the imaging time is going to also go up. So it's not a total free lunch. And then to the right here, you now see Stefan's new microscope. And there, of course, he is pointing at the donut. Even you maybe think he switched from microscopy to work in the pastry industry or something like that. So, but now using the donut as excitation source, you can then pinpoint individual molecules. Let's see if I can replay this video. So then combining single molecule blinking with this donut to track where the coordinates are, then you can improve the precision compared to single molecule localization microscopy down to a few nanometers. And we actually installed this system about a year ago at SciLife Labs. So we are also supporting projects with main flux microscopy. And then this is my last slide. So I just talked about methods, kind of how the methods work and things like that. I haven't mentioned much about fluorescence molecules and things like that. But the methods that you saw, in principle, they work for almost most common dyes that you're using. And then, of course, regarding sample preparation, I haven't talked at all. That's probably what I consider to be the most advanced method in fluorescence microscopy to really get the sample as it should be. And then, of course, you always have to ask your question, why do I want this image? What am I after? What parameter do I want to measure? And that needs to be planned in your project. And then, of course, I also not cover anything regarding image analysis, but you get that in the whole course. So that was my lecture.
Hey, thank you very much. Nice to get this very big overview. We actually have quite a lot of people this year who use we have one student who actually uses Eriscan for everything, and then we have several students who have work on the system with Eriscan, but actually don't know what it does or have never tried using it. So I think that was very useful. You guys tell us what you think about the lecture, and if you have any questions about your own sample. Some of you really need a high resolution, and many of you use high resolution and don't need it. So what are your thoughts?
I think everybody is hungry. They want lunch.
Then
Yeah, actually, I haven't used that before. Yeah, basically, the size of object and distance of objective in my samples, seems don't need high resolution, super resolution microscopes. But I'm curious about that. For example, if we want to use that, how long is the procedure of sample preparation is the same at the normal sample?
Yeah, all the steps are in principle the same. Immunostaining, and then you just try to get the sample as good as possible. So maybe I would recommend, I don't know, are you looking in cellular samples or what are you looking at?
Yeah, it's a cell sample.
Yeah, okay. So of course you need to maybe avoid growing your cells in these what is it called? Phenol red, if you want to push it to the single molecule regime, because
Okay.
every step in your protocol that can increase background is going to blind you when you go to the highest resolution. So just look up at the sky and tell me how many stars do you see. If you can turn down that background, you're going to start seeing the molecules. So usually some tweaking on the sample preparation is kind of needed. The first thing we also want to see is probably your confocal images of your cell, how it looks like, or your wide field image, so we can tell if it's going to work or not. But otherwise, you just follow your normal protocol. And I prefer, of course, super fixed, flat, super bright cells in order to benefit from the highest resolutions. Because I also told you that instead you only see a few molecules, and of course if you blink individual molecules, you're a bit contrast limited there.
Thank you.
and then there are some. I mean, it's for for everything else the same. But I think we spoke a lot about sample preparation in this course. And, sometimes I say sometimes you can get away with murder and sometimes you can't. And then when you can't, then it's about time to just look at all the tips that we have given and follow them, and really it makes a big difference. And this is the type of things that you see in super resolution microscopy. So super resolution microscopy is a little bit like when you use a low NA or a high NA objective. With a high NA objective, you're going to see a lot more of all the problems. And then in super resolution microscopy, it's even worse. So this is when, if you have good habits in sample preparation, then it's you're on the right side already, or the right track.
Yeah, usually we just take a confocal sample if that's good, and we just put it on the STED microscope or the SIM microscope, and you would have the results right away. And then from that image, we can of course see if something needs to be tweaked, or we recommend maybe shifting some dyes to a different spectra or something like that. So yeah, it's usually quite simple if you have the good sample.
Many of you use, oh yeah, Laura. Yeah, Laura needs high resolution.
Yeah. I have a question actually. It's there are like better wet lines that it's going to be better for some super resolution. Like the green ones is better or something like that.
Okay, yeah, that's a very good question. Of course, you learned in this optics course that resolution scales with the wavelength. So in principle, we should do all the imaging in UV because that's the shortest wavelength, because that has the highest resolution. That was actually 100 years ago where they actually developed the first kind of like 200 nanometer wide field resolution microscope. But as you also know, low wavelengths generates a lot of background. The glass in the objectives are maybe distorting this wavelengths worse and things like that. So you kill your cells. So usually we recommend that you actually go to the other part of the spectra for the highest resolution, because there your background is much lower. So we usually are up in the red, far red, with STED and single molecule localization microscopy. And as long as we get a lot of photons out, a lot of nice contrast, we don't care that the wavelength has become slightly larger, because the method of fitting, so to speak, the profiling single molecules scales a little bit with the wavelength. But if you're above the background and have good signal, that's a winner. And also for STED, if you sit on low backgrounds, you can actually use far red depletion wavelengths, because the STED laser is quite intense. But if you're then up in the far red, you don't kill your sample as much as you would do with UV lights. So actually, you should avoid even blue excitation on live experiments, even though of course the resolution is better in depth.
Oh, and for fixing samples, so it's better for me don't image it, don't stain the depth or something like that.
But of course, depends on your question. There are of course also 500 different DNA dyes, and several are actually in the orange or in the far red. So it depends on how you plan your experiments. Of course, multicolor is always going to be problems, because tweaking spectra and crosstalk and overlap and things like that, it takes a while. The trend in modern microscopy right now is to continue up into the near infrared. So you then select dyes that are even above 700. You can of course not see them with your eyes, but the detectors can see them. So then you buy another spectra in your microscope up in the far red.
Okay.
So,
Thank
but for screening purposes, of course, nice to have the dye in green, because your eyes detect green the best. So it also needs to be planned there. What is your morphological channel where you, so to speak, say, okay, this is the cell type I want to look at. So But maybe don't put super resolution in that channel. You then take that in the orange or far red or something.
Okay, thank you.
So we spoke a lot during the course about the signal to noise and the signal background ratio and the sources of noise and sources of background, and how we can how actually increasing the signal is decreasing the background and the noise is as useful as increasing the signal for the contrast in the image. So we said a lot about that, and I think that Laura has works on very small leishmania. They are very tiny things, parasite, and the small vesicles inside. So
Okay.
there we spoke
Well, it
about.
should be possible. We looked at bacteria, small bacteria, pneumococci, and we look at these all these what I called soap bubbles in the cell, vesicles and things like that. So it's possible to look at them with a little bit better resolution. Of course, if everything is super 3D clumped, and of course you would need to make a new microscope. You need a new Nobel Prize. That's like a few nanometer 3D resolution, and maybe that is then the Minflux method, which is of course not easy to run and use, but we are getting there. We're becoming more and more experts on that method too.
And I was wondering, is there any so Lara is actually in Brazil. Is there any possibility that she sends some sample, and can you accept things that are outside of Europe? Because I know that you can image things from anywhere in Europe, right? But
yeah, there is this bridging pan-European network called Eurobioimaging. And I think we also have a connection to the whole global bioimaging community. So you can probably just go through those network and then ask for support, and then we will pick that project up and have project discussion, things like that. So that's possible.
Yeah, this is great. It's a fantastic support we have here,
Yeah.
So if you get a chance,
Yeah, and then as I didn't cover this expansion microscopy method that was invented by Ed Boyden 10 years ago, that has been tried on many things, even bacteria, plant cells.
Yeah.
So you could look up some of those protocols and try to get your sample a bit stretched. And then you can actually use formal confocal microscopy with the 4 to 10 times stretched sample, and then you bought 4 to 10 times higher resolutions.
Yeah, I tried the expansion microscopy actually. We have the protocols and we try it, but I need to see pores in the membrane. So I don't know if the membrane is actually going to be affected, but
some protocols, of course, then you wash away the membranes, because the most things that are scattering in the cell are all those lipids that generate what I called soap bubble layers. But maybe there are some protocol out there that have tried it on some bacteria that you can use or something. Or you need to invent a new protocol, and then you are the winner, because the best sample preparation is always going to be the winner, because you can find a microscope wherever. You just take your sample to a microscope.
Laura, you are labeling the pores. So you label the pores with an antibody. So then there are some proteins that are specific to these pores. And even if the membrane disappears, you can visualize the proteins. So it should work.
Yeah, you can fix the protein onto the hydrogel or you fix them already in the first step. So if you are after the protein topology, then that should be possible, even if you lose the membranes. And there was somebody in the chat here that
Augustine, who says that you can go to LABI, Latin America Bioimaging.
Okay, nice.
Very nice. And you can do super resolution in Argentina. Yeah, Augustine is from Argentina.
Okay.
Nice.
I also want to go to Argentina. So.
Oh well then. Yes, Sadia.
I have a question. So if I am checking like a bacteria and host cell interaction, so in this case, would super resolution microscopy be a good idea? Just for screening for microbes, which are
Screening, I mean, you need to all your screening with more conventional method because
okay.
super resolution is no high throughput. It's high
Okay.
high resolution.
Okay.
But
High
if
resolution.
we then want an image on your surface and the topology on the surface, then of course we can try that.
But for example, if we want to see if it's causing like DNA damage or something more specific, or there is a
Yeah, but now you know that the DNA is really nanoscale. I mean, it's like
okay.
1-2 nanometer spacing between double stranded DNA, and then that's curled up in like 3D structures that are just tens of nanometers. So there, the super resolution is kind of failing because it's too small and too compact. There is, of course, a possibility maybe to measure something that is not too small, maybe protein assemblies onto DNA structures and things like that. It can't be too crowded. Of course, you need to label this very, very nice and specific. As soon as you go into the nucleus and you try to label things, you usually label all the nanospaghetti, as I call it. So there I would say that again, the sample preparation is more important than actually the microscopy method to get that under control. No unspecific staining, no background. But in principle, we should be able to crisp up any microscopy image with these methods. But if it gets too small, that's why I also wrote there above 25 nanometers. And as soon as you try to go below that, then you're you need a lot of work. So there are crazy ideas, maybe combining expansion microscopy with even the worst super resolution minflux methods and things like that. So
And also, Sadia, so if you have a screen, what you can do is do a pre-screen for it with another method and then get some candidates and then look at the mechanism in super resolution microscopy with a few candidates instead of doing the screen with super resolution is not really possible. But, if you do a pre-screen and you have some candidates, then that works.
Yeah, that's the way it should work because
All right.
as Sylvia said in the beginning, some of you are actually using the wrong method for your questions. So wide field microscopy still has 240 nanometer resolution and then a little bit of image processing and blah, blah, blah. I mean, those are small numbers. Go home and tell your parents to explain 200 nanometer to some. It's a very small number. Now, of course, if I can help with another factor of 2 or 4 or 5 or whatever, those 50 nanometer or 20 nanometers, they are extremely small numbers. But biology is even smaller than that, unfortunately.
True. Thank you.
More questions for Hans? Good, we have a good discussion going, so I'm happy.
I don't know, there was some quiz questions that you were
Yeah, we have some
supposed to
quiz questions.
answer.
You want to do the quiz together with us because
I looked at them and of course in microscopy there's no right or wrong answer. It all depends on for what and here I only talked about the resolution parameter. I know there are
so if you guys
time
go
and
to
live and blah blah blah.
If you guys go to modules, we can do that here. And to go to modules, and then I can share my screen, and then you go to this super resolution microscopy quiz. Okay, and then you try to answer. Then we can discuss the questions together. And after that, we will go to the other Zoom to do the other quizzes.
By the other quizzes, you then mean the questions.
So we have
you
lots
shared,
of quizzes in the course. Am I sharing? You see my screen, right?
Yeah, but you also send me some other questions regarding resolution, span of super resolution microscopy, and then
this one, so one should turn to resolution microscopy if the resolution required by the scientific question is between best resolution that a single point confocal can acquire. So what is the best resolution that a single point confocal can acquire? And the best resolution that super resolution can acquire. What is that?
But then you need, of course, to know what wavelength, in what lab.
Yes.
Is it well aligned? For what objective? For what day of the week?
So there's a lot of gray zones in this, but maybe you can pick something from what you have seen in the lecture, and in here you can pick something from what you know. What is the best resolution we can get? And then the next question was about in which way SIM and image scanning microscopy, which, for example, Eriscan that many of you have on your microscope, in which way do they differ from the STED and single molecule localization microscopy techniques in terms of field of view? So is it better? Is it worse? Or does it depend on the technique? The flexibility of the fluorophore choice, the speed, the resolution, the number of fluorophores you can image, and the depth of imaging. Is that really a try? I think that I can see somewhere when people publish, but I am not 100% sure where. Okay. If you have answered, please raise your hand. If you have submitted. I can see that people have submitted. Wait a couple more seconds. Maybe we have a quick look at the answers. So I'm just going to do it this way. So when you submit, you will get the answers here. And yes, for the what is the high and low, it doesn't look very nice. I'm going to just submit, and you will see the way you guys do it. So just do it this way. So let's see, if I just select the first one for each of them, and then I'll have everything wrong, but at least it will go a bit quick. Then we will see the answers. Submit. So now I did lots of things very wrong. So what was the single point confocal? What can the single point confocal acquire? What type of resolution? 300, 200 to 300 nanometers would be the best. It depends on the wavelength, as you say, it depends on the sample. And if we look at the super resolution microscopy, what is the best? that one can answer. The correct answer is about 20 nanometers. So
yeah, but that's extreme.
Extreme 25. Oh, now we're like so something like this, 20, 25. So between around 200 and 200, I think is also quite extreme.
Yeah.
So we'd say 225 to 250. That's about there, where you can use super resolution microscopy. And then what happens to the field of view? So the field of view is when we look at the techniques that are SIM and ISM, so what Hans called soft super resolution microscopy, and compared to the hard ones, I guess, in that case, then what we have a field of view. The field of view is better for SIM and ISM compared to STED and single molecular microscopy. And then the flexibility of choice of fluorophore is also better for those. Basically, you're not limited, but clearly you would want to anyway use the best ones, whereas in here you have some constraints. And the speed of imaging is also better. However, clearly the resolution for these hard ones, these hard techniques of super resolution is much better. So we have a difference in maybe 10 times or a little bit less than 10 times, maybe 5 times. So better resolution in some techniques X, Y, and Z. And then the number of fluorophores in these techniques, you can often image 5 up to 6 fluorophores, but in here you're maybe limited to 3. I guess, and again, it's difficult. But then the depth of imaging is also much. In some cases, you can go deeper in here compared to here. So I think that when you choose if you're going to use super resolution microscopy or not, and which type of technique, you have to consider all these things and see what is the best
Regarding depth, you can always kind of generalize saying a confocal pinhole-based method will help you as it has a physical depth rejection. And as STED is a confocal method, it's actually easier to go into depth than, for example, SIM. And of course, single molecule localization microscopy is also camera-based. It's of course super hard to go into depth. So the pinhole will always help you if you go into depth. And of course, if you want to go really, really thick, then of course you need to switch to light sheet illumination, where you have excitation in one different direction compared to your detection directions.
Are there any questions? Those people who have their hands up, is it because of question or is it because to say that you have finished the quiz?
It's to say that they are hungry and want to go for lunch.
They're hungry. No, we have some quizzes before. So we're going to move to the other Zoom now. Hans, I want to thank you very much. Chen, did you have a question?
No, I just forget to.
Okay. So I want to thank you very much, Hans, for this very nice overview of super resolution microscopy, and I hope that you get lots of people coming to you with great questions and good samples.
Yeah, okay. Thank you.
Okay,
Bye-bye.
bye-bye. Have a good day. So everybody else, we go to the other Zoom now.